Two sets of stainless steel wires are cut at different lengths, prepared and placed into the protective plastic tubing. Once the wires are prepared, wiring of the crayfish consists of drilling small holes in the gill chamber to span the sphygmomanocyte as well as the thoracic chamber where the wires span the heart. The wires are placed just under the cuticle and sealed with adhesive glue. These wires are then connected to an impedance detector which measures dynamic resistance between the wires in each set. The impedance detector is connected to an analog to digital board, which is connected to a computer. Software provides the gathered data as waveforms that can be collected as beats per minute format for experimental analysis.
Hi, I'm Sonya Bierbower from the laboratory of Dr. Robin Cooper in the Department of Biology at the University of Kentucky. Today we will be showing you how to record electrocardiograms and electrosphygmomanogram in crayfish. We use this procedure in our laboratory to record the autonomic response in crayfish in varying environmental conditions. So let's get started.
To begin preparing the recording wires, cut two insulated stainless steel wires to a length long enough to be inserted into the crayfish and adequately reach an impedance detector. Use a flame to remove about 0.5 mm of the insulation on the ends of both wires, which will be inserted into the carapace. Be careful not to remove too much insulation, as a bare wire on the outside of the animal must be recoated to prevent short-circuiting. Next, use forceps to bend the burnt ends of the wires at a 90-degree angle for placement into the carapace. Make sure that the inserted portion of the wire is not too long, as this could damage internal organs. If you are recording both heart and ventilation rates, repeat these